we start, or should we?
Yeah, no, I think we should start.
Okay, so yesterday we were talking about the evidence-based approach towards classification of variants that are identified in sequencing reactions. This could be any sequencing reaction. This could be a next-gen test, or this could be a Sanger test for a single gene. It doesn't matter. In the end, you're still looking at DNA variants, changes in the patient or the sample that you're looking at versus the reference material. And I told you that this is an evidence-based approach using these pieces of evidence, population, frequency, type of mutation. So if you have a variant that, say, is at 5% or greater in the general population, you can safely say that that is a benign variant. You don't need any other pieces of evidence for that. I'll talk about that a little bit later. So these levels of evidence are broken up into either towards pathogenicity or towards being benign. And then there are different rules. So this is rule PBS1, a null variant, non-zest frame shift or canonical splice site change in a gene where loss of function is a known mechanism of disease. And I talked about an example in BRCA1 yesterday. You sequence someone who wants to know if they have a predisposition to breast cancer, and you find a loss of function mutation. Those are known in BRCA1 already, so you can safely classify that variant as likely pathogenic. And if there are more levels of evidence that add up, you could even move that up to pathogenic. So you take, and for every variant, you look at each one of these rules to determine yes, no, or not applicable. Then you add it all up using this rubric. So if you have one PBS1, which is the rule I just talked about, and one strong rule, which I didn't talk about, you can safely classify that variant as pathogenic. And on the other side, so these are the P for pathogenic scoring criteria. On the other side, there's some Bs. So if you have a BA1, which is greater than 5% of the population, you can safely classify that as benign. This is a better way to show this. This is a table that shows you the levels of evidence. So if you're over here, the minor allele frequency is too high for the disorder, that's strong evidence towards being benign. And again, if you have a predicted null variant in a gene where loss of function is a known mechanism of disease, you have very strong evidence towards pathogenicity. So what this is doing is it's trying to put levels of evidence on these variants so that if you get a variant and you get a variant and you go out to determine whether or not it's pathogenic, you come back with the same answer. And the reason this was written was because what happened when NGS hit the clinical labs a few years ago is people weren't giving quite the same classifications or using the same terminology. Some people were saying disease-causing, some people were saying pathogenic, some people were saying affected. So this makes the community use all the same language and try and come up with the same scoring. It's really complicated, and you do get different scores, slightly different from different labs, but for the most part it's working fairly well. Does that make sense? Okay. So this is just an overview of the sequence variants that you can get. If you get Sanger sequencing, say either doing the targeted panel or targeted mutations or the whole gene, you're going to get ones of variants. When you get to the whole genome, you're going to get thousands of variants. So the time for analysis and interpretation, which I told you adds money to the test because you're adding hours and hours of work, goes from minutes, maybe for a panel of some 23 variants, to hours to days, depending on the whole genome sequence. And the whole goal is to get these variants from ones to millions. Each one needs to be in one of these categories. The category that the majority of things fall in for whole exome and whole genome is the variant of unknown significance. And this is important because you're trying to give a patient an answers to what they have. And if you're out looking in their literature, you're looking in the population frequency, you're looking for information to help you score, but there just isn't anything, you have to give back a variant of unknown significance. You don't know what it does. Hopefully in the next few years, as we collect these data over and over and over and a lot more people, the number of VUSs that we will report will go down. And I'll show you that that's actually, go ahead.
So is there some type of database where you guys enter the variants of unknown significance?
I'll show out, yeah.
Okay.
So let's talk about multi-gene sequencing panels. So we talked about targeted variant, we talked about sequencing single genes, now we talked a little bit about annotation. Now let's talk about doing a whole panel. This is the basis for the whole exome. So I'm not going to talk about exome because it's too complicated to get into how we score it in the short period of time that we have. But we can use these gene panels as an example. Because a gene panel is just a small, tiny little exome if you think about it. So sequencing panels are used for conditions that can be caused by changes in different genes. So this is like the muscular dystrophies that I talked about. There's a whole bunch of different kinds of, there's a whole bunch of different genes that cause the same disease, right? Osteogenesis imperfecta is another. Noonan syndrome, hearing loss, is a broader phenotype that if you have hearing loss dependent on the kind, conductive or sensorineural, you're going to test a separate panel of genes. But there's some defined number of genes that you look at. So let's talk about the different types of muscular dystrophy. And I'm just going to go through this really quick in the interest of time. This is Duchenne and Becker. The affected areas are indicated in the gray. So the upper limbs, the upper arms, and the upper legs. This is the Embry-Dreyfus type. It just affects the upper arms. You can see the pattern is a little bit different of which muscles are affected. We're going to talk about limb girdle today. So that's the upper legs, the upper arms, and then the pecs. And there's one here called, this is an interesting one because it's called oculopharyngeal. It has the same patterning, a little bit overlap of limb girdle, and facioscapular, but it affects the muscles of the eyes as well. So these are the different kinds. The X-links are Duchenne and Becker, which are both caused by the DMD gene, and then the autosomal forms are limb girdle, congenital, and the others down here. So let's talk a little bit about limb girdle muscular dystrophy. So again, let's look back where we're looking at the affected tissues, or affected muscles here. It's a generic term devised in the 1950s to delineate the autosomal forms from the X-linked forms. But we've moved on from that now. We have a whole bunch of other forms. It's a heterogeneous group of diseases with muscle weakness in the shoulder, pelvic girdles, and the upper limbs. That's fine. It's variable in the age of onset, and there are autosomal recessive and autosomal dominant forms. No facial involvement, normal intellect, sometimes cardiac involvement. Again, elevated CK in the cases because you have that muscle breakdown and the CK is being released into the blood. And immunostaining can help direct the genetic diagnosis, but you do need to do genetics to figure out which gene is being affected, to calculate recurrence risk, and other things like that. And these are just examples of the immunostaining here. You can see this is one of the genes, the muscle fibers. I don't remember, oh, this is dysferlin. Being stained for. You can see this is positive, but in a person who's affected, there's no dysferlin. So this is a great test. You wouldn't even have to do genetics. You know that this gene's already involved. You don't know what kind of mutation is there, but you would know the gene that's involved. And these two things here are just showing you the difference between the affected muscles and the unaffected.
They always take a muscle biopsy?
Not always.
Okay.
And I'm going to tell you why. This, you might know that dysferlin is affected, but these are all the genes that can cause limb girdle muscular dystrophy. So you're not going to take a muscle biopsy and do 50 different immunostains. Why? Well, it's expensive. B, why? One, it's expensive. Two, we know immunostains probably don't work for all these. You know, that's a tricky test sometimes. So what do? We look at genetics. The other thing I want to mention is that limb girdle muscular dystrophy genes overlap some of the other muscular dystrophies as well. So you may see that some of the genes are in multiple panels. Okay, this is a test from 2014 for limb girdle. It includes the autosomal dominant and autosomal recessive genes. You do not need to know the genes. This is just telling you what they are, the number of exons, and what type of muscular dystrophy that it causes. This is like hearing loss. There's special codes, LG, MPL2, just to tell you if it's autosomal dominant or autosomal recessive, but don't worry about that. The key point here is that this is a 24-gene test, testing 335 exons. So if we wanted to do this as a Sanger panel, we'd have to pipette 335 reactions for the patient, 335 reactions for water, 335 reactions for a positive, do the PCR, run a gel, make sure all the bands are there, clean it up, throw it on the sequencer. It doesn't, it sounds like a lot of work, but and it is, but comparatively, in the wet lab, it's the same amount of time as NGS, but the NGS analysis is a lot faster. So we can look at all these genes now, all these exons at the same time. Okay. So let's talk about, and this is where the databases come in, let's talk about the first level automated filtering. So you take your 335 exons, you do your NGS, everything passes the quality that we need to look at in the clinical lab to make sure that we can report it and use the data. And that's important. If something fails, we stop. We rerun. We re-extract DNA, we start over. Let's say the 335 exons look good. The first thing you're going to do is go to dbSNP, which I'm sure you're all familiar with, the exome sequencing project, which is was a project that was started about 5-6 years ago to sequence, it was about 8,000 people, which at the time was a ton of exomes. You look against the 1,000 Genomes Project, which started at 1,000 exomes, and now we have EXACT, which is the Exome Aggregation Consortium, which is a collection of 60,000 exomes from all around the world with 6 different subpopulations, and that's important for screening of these benign variants. And then you may have an internal database within your lab. So Baylor's been doing exome and sequencing for a long, long time, so they have their own internal database that no, they don't share. Myriad's a good example. They're not going to, they've been doing BRCA1 testing since they patented it. So they have a proprietary database from a long time with many, many mutations that maybe they've only seen. So one thing I do want to say is the EXACT database is being replaced already. They just published a paper in August explaining the database, but now they have 120,000 exomes, and they're changing the name to Nomad. So just something to keep in mind. And I don't have time to show you these databases, but okay, awesome, perfect. So any variant present in greater than 5% in the general population, and that's any subpopulation. So if you see in East Asian Islanders that they carry a variant at 5%, you can take that out as disease causing. It's very, very unlikely that in Mendelian disease you're going to have a variant that high in the population that's pathogenic. There are some caveats to that. The hereditary hemochromatosis gene, something like, I don't know, 23% of Caucasians carry one of the variants. But that's different because that disease isn't 100% penetrant. So these are things you have to think about when you're designing your test too, is the penetrance of the disease versus the filtering that you're going to do. But in general, 5% is a safe cutoff. And then actually, variants greater than 1% are also filtered out, but not automatically in all cases. That's where you need someone who's been trained and who makes a directed decision. Yeah, go ahead and toss those out. For cancer, anything over 1%, we toss out. If there would just be too many people walking around with cancer for that to be feasible. And that's what I'll say about that. Does that answer your question? So these are the data from the limb-girdle case that I did in 2014. So again, 335 exons, and we only identified 49 DNA changes versus the reference sequence and our patient. 49. 45 of those were automatically filtered out based on population frequency or whether or not we had already seen them and classified it in the lab as benign. Because this was a really big reference lab that I was working in. So that only left three variants that actually the technologists running the test classified them as benign on their own, and then they got to the director for further classification. And I agreed, I said, yeah, those are benign, so we put them in the benign category. And that just left one variant left, in a gene called ANO5, and it was a C1442 T to C, and I know that nomenclature has already been gone over. So how do we determine whether this is pathogenic or not? Well, to give you a shortcut, we'd already seen this in the lab, we knew that it was pathogenic. But if we hadn't, we'd go into different databases to look at that. And one of them is HGMD, the Human Gene Mutation Database. It's now presented by QIAGEN because it's been bought out. It was started in the United Kingdom, and about 6 years ago, this was an amazing database to have. They had teams of people reading papers, and curating any variant they could find. It was at the time, it was the best thing you could have, but it cost $28,000 a year. So only the big reference labs could afford to do that because you have to cover your cost of your test, right? So they could dilute that $28,000 over hundreds of thousands of tests. In our lab, we might run thousands of tests, small thousands. So to dilute that, that's a lot of money that gets added into the cost of the test. Academic access is free, but limited. So you can use it, and it'll tell you that that there's an entry in the database, but it won't tell you what it is. This is just a screenshot of the database. This is an old slide. So limited access equals limited utility in this case if you're using the free version. It did tell me that C.1295 C to G homozygously was in there, but it wouldn't tell me the classification. So the answer to this on the public side was that there's a few people up in Boston and in Pennsylvania who have really spearheaded something called ClinVar. And I don't know, did you go over ClinVar? Okay. So ClinVar is the public answer to HGMD. It's where the clinical labs, if they agree, deposit their information about the classifications. So each entry is scored based on a star system. So if you take like this CFTR variant for cystic fibrosis, is proline 79 serine, and cystic fibrosis, it was classified as a variant of unknown significance. This is an old entry, and it has 1 star. So 1 star means it was classified by one person who dumped that into the database. It's not been vetted or charactered or checked, but it'll tell you who submitted it. So that way you know a little bit about the lab. 2 stars means it's been classified by multiple submitters. Let's say GeneDx put it in there, Invitae put it in there, Baylor put it in there, then you're going to get 2 stars. 3 stars means it's been reviewed by an expert panel. So, there's societies for melanoma, and there's societies who solely study cancer in general, who have work groups for certain diseases. Let's say they've looked and they go, oh my gosh, this is disease causing, we agree, that one gets 3 stars. 4 stars is reviewed by a professional society, so that would be like the ACMG taking it on and going, we agree. I've never seen anything with 4 stars. So if you look in ClinVar for the mutation that we have, this AN05 mutation, this is really small, but it's pathogenic by a single submitter. So that's one piece of evidence. And this is the entry showing you. This has all the information about the coordinates of the variant, the accession number, which is really, really, really important in clinical diagnostics. The accession number is like the address, right? So if you say C.4, you need to have the accession number because C.4 may not be the same in 2 different alternatively transcribed messages of the same gene. I want to go back really quickly to, this has sort of changed recently since those guidelines have come out. The scoring criteria for the public databases for pathogenicity is now 1 point in that scoring rubric. So if you find that it's pathogenic by GeneDx, you can't just assume it's pathogenic. That adds a point to your levels of evidence. You can't just put something on a report and say, well, GeneDx said it was pathogenic, so it must be pathogenic, because they're really good at what they do. It's up to you to find the reasons why GeneDx classified it as pathogenic. Does that make sense? In court, that would be called hearsay, right? So that's why you have to be careful. You've got to do your homework. Just because somebody called something pathogenic doesn't mean it is. Okay. The other thing we use is just a simple PubMed search, which is now turned into a Google search, honestly. We type in AN05, the C dot number, the P dot number, and we look to see if it's in any paper. And generally, if it is, you'll find it, even if it's buried somewhere in a table or in a supplement, which is what ends up happening a lot of times for benigns specifically. We'll find somebody published a paper and they say, yep, we found this in 3 people with this disease in this gene, and we believe it's benign. You can use that level of evidence. So our mutation quickly is actually due to a couple founder mutations, which results in an alanine to a glycine, and then it frame shifts to 49. This is just a picture of the old version. This is the software we use in the lab, just showing the homozygosity. I'm sure if you haven't by now, at some point in your career, you'll be looking at the raw data for next-gen sequencing. This is just what it looks like. And this is the summary of the results. When you give a report to a patient, you always put a summary first. Positive, disease-causing, something like that. Generally, I like the word positive test, because the physician opens the report and the first thing they look at is, yes or no. So we put positive, we put negative. And then a small results in interpretation. So this patient is apparently homozygous in the AN05 gene for a variant defined as this, predicted to result in amino acid substitution this, previously reported, and then the reference. So why do we say apparently homozygous? I mean, look, every single read is a variant, so it's homozygous. Why in the report do we have to say apparently? What happens if there's a deletion on the other allele? All we've done is PCR and sequencing. If the DNA is gone on the other allele, we won't know, because we stop there to PCR and sequence. Or if there's a rare SNP buried under one of your primers, you're going to get allele dropout. So we have to say apparently because who knows, the patient goes on for further testing and they find a deletion on the other allele, you're on the hook. And then predicted to result in the following amino acid substitution, unless somebody's actually looked at the protein and shown that this is what happens. All you're doing is predicting, and that's sort of a key thing. So in a similar vein, let's say you have your exon and you're out +1, +2, and you find a DNA change. That's going to affect splicing, right? Because you know about canonical splicing. So you're probably going to read into the intron and frame shift. But do you have proof of that based on DNA? No. Go ahead.
I just was wondering if you guys ever look into like if it's a variant that there's not a lot of information about, like whether it falls in a domain region or if it's a conserved region, like would you write that up?
That's all part of the scoring rubric. So whether it's conserved or whether it's in a known domain function, that adds to the score. But if you're still in a VUS, the VUSs just get listed as a table. But if it's likely pathogenic, if it's hit that threshold of likely pathogenic, we would say something like it's in this domain, or something like that.
Yeah.
This is a really short write-up. So they tend to be a little bit longer.
Okay.
The person I work with like to be very succinct. And I'll be honest with you, physicians open the report. If it's a physician, they open the report and go, oh, got it. They don't read. Genetic counselors read and synthesize. So, but that's it's the difference between someone who sees a patient for 15 minutes, someone who sees a patient for an hour. And I'm talking about specialists. I'm talking about if a GP orders this, which they rarely do. But more and more as genetics is becoming more mainstream, you're getting GPs ordering genetic tests. We're getting weird doctors ordering them. Let me call them. Do you really want this? Should you call a genetic counselor? I had an OBGYN who should know about this stuff order a Fragile X, and I called out the result. I said, you know this is positive. He goes, I don't know what to do. And I'm like, why don't you refer them to genetic counseling? So, okay. Any other questions? All right. Like I said, the whole exome sequencing, it works the same way. You get a VCF file, you filter, and you try and go down from 20,000 or more variants to a handful that you need to analyze. And as people are now communicating and making these publicly available databases as the exact and now Nomad databases are online, we are finding out that the majority of DNA changes are polymorphic, are in the population at some level. Now you have to be a little bit careful, and this is a big red flag. Population databases contain unhealthy people at some level. Maybe they haven't developed the disease, maybe they didn't report it on the sheet they were filling out. You know, do you have cataracts? Yeah, but they forget to check the yes button. So you just have to take those with a little bit of a grain of salt. But the filtering, the number of variants that we're having to analyze is going down. All right. Whole exome sequencing. This is really important. It's not a first-line test. You're not going to get a kid walking into the clinic, have a meeting with the parents and the child and go, let's do your exome. There's going to be metabolic tests first, there's going to be blood work, there's going to be single gene sequencing, targeted. They're going to try and determine the genetic cause of the disease generally first. And that's because it's a relatively expensive test that insurance will not cover, although that's changing too. We're really in flux right now with all this. The cost is dramatically decreasing. I thought I would. Decreasing, and it's becoming competitive with panels, which is why in the wet lab especially you're seeing the exome, whole exome being sequenced and bioinformatically masked, because it's just as cheap to do that as to do a panel.
How much does a full exome sequence cost for like a patient?
I think it's about $3,000, $4,000. The other thing you have to remember is there's list price, there's what the insurance pays, there's what the patient pays if the company has a special agreement. There are companies out there, and I'm on tape, I should be careful. I don't agree with pricing structure like that. I don't agree that a company should say, well, if you don't have insurance, you're only going to pay $100 out of pocket. If you do have insurance, you're going to be billed this much, and don't have the right insurance, or your insurance doesn't work with us, the price should be the price, and the insurance should pay what they pay, and you know, if they don't have insurance, there should be a payment plan. I mean, this structured hidden pricing is, and this is just our healthcare system, unfortunately. And I get a bill from the hospital here for $28,000. My insurance paid them like $6,000. If I didn't have insurance, I would be on the hook for $28,000. So, anyway, let me hop off my soapbox. The coding section of the exome is 1 to 2% of the total genome. It's 30 megabases. So that's why we can sequence it so easy. But 85% of the known disease-causing genes are located in the exons. So that's why we don't do genome sequencing in the clinic yet. You guys in the research lab, please, do whole genome. Let's figure out what's going on. But we can't do it. We just can't do it. Obviously, a large amount of data are generated, and you need a really robust bioinformatic pipeline to handle. That little panel with 335 exons, we could have probably done that on an Excel sheet and checked them all off one by one. At 20,000 variants, you need computing power to do that. All potentially positive changes, meaning pathogenic or likely pathogenic, need confirmation with Sanger sequencing. This is sort of a holdover for when NGS hit the clinic a couple of years ago. People didn't trust it. Even if you had 200 reads and 50 were wild type and 50 were variant, they still Sanger sequenced because that was the gold standard that everybody was used to for years and years and years. People like to see that little peak on peak for verification. As we're finding out that the next gen is robust enough, that's not as big of a concern anymore. Many big labs are not Sanger confirming. As we go online, we will Sanger confirm until we're comfortable that we're catching everything. If we find a variant after a few months, all our variants are Sanger confirmed at 100%, and we'll just trust the data. Alright. So when would you consider an exome? I told you it's not a first-line test. So in a phenotype or a family where the history suggests that genetic etiology but a specific genetic test is not available, that would be a first-line test. If you can't find anything else, you could do an exome. It's really rare that you wouldn't try something. A defined genetic disorder with a high degree of genetic heterogeneity, making it a more practical approach, especially as the price decreases. So instead of doing a panel of 172 hearing loss genes, maybe you do throw an exome at it. That's a bad example, because hearing loss, you do a panel. This is especially true as the price decreases. And then this is the important one. Patients with likely genetic disorders, but a specific genetic test have failed to arrive at a diagnosis. These people are on what's known as a diagnostic odyssey. And I don't know if you've heard this term before, some of you probably have, you'll hear it a lot in the future. If you have like a 12-year-old with intellectual disability who has not developed well for years and years and years with developmental delay and learning problems, they've had every test under the sun. They've been in and out of neurologists and physical therapy and pediatricians and endocrinologists, and they still are at square one. This is when you have an exome. I need to skip ahead. Okay, perfect. Alright, so how do you go from 20,000 variants again to the pathogenic variant or variants? There are people out there who have more than one genetic disease. That's something to remember. It's much as described about the panel like we did above, but just on a much, much larger scale. Much more computing power is needed. And it takes a little bit longer because in the end, after you've done all the automatic filtering, there still are a bunch of variants that can't be classified automatically. And you do want to take a grain of salt on things that are classified automatically. That's a computer doing the job. Human eyes should go over as much as possible to make sure that you're not missing anything. And again, the things that are included in that are the type of mutation, the presence in controls, the in silico data. This is not weighted very heavily, and that's good. Conservation, hotspots, domains, functional analysis. With panels, they're inherently phenotype driven because all the genes cause the disease you're searching for. So you don't take phenotype into account when you do the variant analysis. In exome, it's phenotype driven. You have so many variants that you have to use phenotype filtering. If you haven't heard of the human phenotype ontology, you should look that up. It's not on the test. but it's something that there are specific terms. It's a website and a consortium. There are specific terms that you can use. So again, trying to standardize things, like instead of saying mental retardation, because we don't use those words anymore, but you might have somebody who tries to use that. We use intellectual disability. And there are different categories of intellectual disability. And you see a theme. We're trying to standardize everything so that we all speak the same language. Just like using CDOT numbers with phenotype, we all want to describe somebody with Noonan syndrome in the same way so that we can get the same variants to be pathogenic, and or not get, score the same variants to be pathogenic or benign. Something that helps tremendously in your variant analysis is a trio. When you sequence the proband and the parent. Now, why is that?
Because if you have a VUS in the proband and you test his parents and either one of them has it, you can say it's likely pathogenic, but if they don't have it, it would be not.
Yeah, depending on the inheritance pattern, right, too. I mean, maybe that's something else that you have, that I didn't mention, you have to know. If it's autosomal dominant and it's one of the parents has it, it's likely to be benign.
If it's like if it's an autosomal recessive where the parents are fine and the child has it, then it's...
Or de novo, that's the important one too, right? If neither parent has a change and the child does and it's loss of function, that's probably what you're looking for. So, geneticists, let's say you have an autosomal dominant condition. The child has it, you find a variant that you think is pathogenic, but one of the parents has it. What could be a reason for that? Yep. Or you get the gold star for today. Yeah, those are things that. So this testing isn't straightforward. These are things that, again, you have to remember. Just because a parent has a variant doesn't mean that it's benign. We had a case when I was in grad school with a kid with Smith-Magenis syndrome, which is highly penetrant and highly, you have it. And the mom, her blood showed she had a deletion, but she was mosaic. So, and it turns out she had a little bit of a learning disability. So these are, remember I said yesterday, before the fire alarm, we don't say in genetics 100%. We don't say always. There's always an exception somewhere. All right. So the results could include a gene associated with a disease phenotype that we already know. You wouldn't hold exome someone for cystic fibrosis, but if you did and you found it, you one-to-one correlation Mendelian disease, you're good to go. You could find a variant in a novel gene, and this is where things get a little bit tricky. In the diagnostic lab, we like to deal with things we know. We like to say A equals A. You have a phenotype, you have a gene, you have the disease. But with exome, this is where things get a little different. This really is an overlap between a clinical test and a research test. So that's why you get consented to run an exome. And you don't get consented to run a CF test. You could also find a variant in a gene unrelated to the disease phenotype, which I'm fairly sure I'm going to talk about it. The causative variant in exome testing is identified in about 30 to 40% of the cases. So that's pretty good. So in 60 to 70%, we still don't know genetically what's going on. So what are some of the reasons why that could be? It could be an intron. It could be something that's not captured well. So I need to move on pretty quickly. I'm taking up a little time. So there are incidental or secondary findings, and this is important. So let's say you're sequencing somebody for intellectual disability and you find out they have a BRCA1 mutation because you're sequencing the whole exome. So now they have a predisposition to cancer. But they didn't ask if they had a predisposition to cancer. So what do?
Well, it's all in the consent process.
It is. But generally you tell them, and I'll talk about that in the second slide. So again, things that predispose to recessive disorders, pharmacogenetic variants, cardiac disease, cancer, you are going to find these things in people occasionally. The other thing that possibly you can find is misattributed paternity. And this is reported to happen in about 3 to 12% of the population. Now it depends on a whole bunch of issues, social issues and other things that I'm not going to get into. But generally a safe number is right in the middle there. About 5% of pregnancies are the dad's not the kid. We see this in chromosomal microarrays a lot because that will show us loss of heterozygosity. So if you have somebody, you run an array, and suddenly 25% of their genome is homozygous, what's the only way that can happen? Incest. And I won't get into that either, but, so there are guidelines. There are guidelines that recommend reporting of incidental findings in exome. So it's 59 genes, and those 59 genes are considered medically actionable. So if you find a BRCA1 in someone in less dystrophy, they get more mammograms if they're female. If they're male, they get more prostate tests now, because BRCA1 now is a prostate gene. Recommended for everybody, regardless of age. So you're going to tell a 13-year-old they have BRCA1, but they're going to have been consented, and the parents are going to have chosen whether or not to get that information. So they can opt out of this. But everybody, almost nobody says, I don't want to know. Because of the genes that are on the list have been selected. Now, lab policies vary in the number of genes. The policy from the American ACMG is 59, but some labs, I think they have a couple hundred things that they consider medically actionable. All right, genome sequence, not widely ordered, maybe in the future. That's all I'm going to say about that. Now, there's one slide about chromosomal microarray, but hint, this is an important slide. This is where molecular genetics and cytogenetics merge. Depending on the lab, chromosomal microarrays are run in the molecular section or in the cytogenetics section. They are designed to look for copy number variants. So see big deletion here, break, that part goes, and this chromosome's a lot smaller. This is different than the exon level array that I talked about yesterday, where we're just looking at single exons. This is looking at the whole chromosome. And it's similar to a karyotype, but with much, much better resolution. So with a karyotype, megabase, 2 megabases, that's what you're going to see changes in. Here, you can get down to about 30 kb. And then anything smaller than that, again, you're going to need an exon level array. Now, because this looks at dosage, you will not detect balanced translocations. If this little piece of DNA flipped over, we wouldn't see that on an array, because it's present. You would see it on a karyotype because the banding pattern would look different. This is recommended as a first-line test in evaluation of individuals with multiple anomalies that are not well delineated. So again, if you're getting to the exome as an order, you've probably already had a microarray and a karyotype. This is really important with non-syndromic developmental delay and intellectual disability. And also for patients with autism, they always get a chromosomal microarray to see if there are deletions. There are specific regions of the genome, like 16p, that are deleted in people with autism. So again, chromosomal microarray, non-syndromic developmental delay, intellectual disability, and autism. All right. So in the last few minutes, let's talk about autism and talk about all the tests that we basically talked about today. So you all know what autism is. It's a group of disorders characterized by impairment in communication, social interaction. It's a spectrum, and it's heterogeneous in cause. It can be deposited to point mutations, deletions, all sorts of things. And without knowing what causes it, there's a 3 to 10% chance of recurrence in a sibling. Okay? So now if we can figure out genetically what's wrong, maybe it's something de novo in the patient, then we can rule out recurrence. Maybe it's something carried in the family. The mom has a translocation, or the dad that's causing rearranging in the child. So here's the recommended workup. Cytogenetic. So you look for chromosomal deletions or duplications, big hunks of DNA that are gone. And this has a yield of about 3%. So 3% of people with autism, you'll find something. So this is only recommended if microarray is not covered by insurance. You're going to want to do a microarray first, because the microarray yield is much, much higher. So if you run a microarray on someone with autism, 8 to 21% of the time you're going to find the cause of their autism. What are you going to find though? Any sequence changes, it's out. Exon level changes, out. Translocations, out. So karyotyping still has, for these people, it's very, very important. And so this is recommended as a first-tier test for everybody with autism. The other test that gets ordered over and over and over is Fragile X, which is what we started the lecture with. Remember, triplet prime PCR, very specific type of mutation. 1 to 5% of males with autism will actually have Fragile X. So again, you have phenotypic overlap of the disease. 20 to 30% of males with Fragile X will have autism. So that's sort of a strange number. You can think about it a little bit longer. Basically, people with Fragile X have autistic tendencies. And so that's just the facts. So testing is recommended for all males with autism. And females could be considered if there are other features or family history. Actually, honestly, I think the physicians and the genetic counselors order Fragile X most of the time on everybody. Okay, MECP2 is another gene which causes Rett syndrome. And again, this has an autistic-like phenotype. 4% of females with autism will have an MECP2 variant. So again, all autistic females are tested for MECP2 mutations. Some small duplications, but it's mostly mutations. So, are you all familiar with Rett syndrome? It's a devastating disease. When I see these pictures of these little girls who were developmentally normal till they're 4 or 5, and then they go through regression, it's really heartbreaking. Again, 6 to 18 months and then beyond of normal development, and then a period of regression, and then things just go badly after that. Now, there are clinical trials for medications that are stopping the Rett syndrome phenotype, and they're working pretty well. So that's really good. And finally, now we've gone through karyotyping and we had triplet prime repeat. Now we're going to talk about a gene called P10. So P10 actually causes a bunch of different disorders. Cowden syndrome is a cancer predisposition syndrome of thyroid, breast, and endometrial tumors. It can cause an overgrowth syndrome. You don't have to remember Banyan, Riley. I don't remember that. I just remember P10 overgrowth. They have a large head, again, a little bit of a cancer predisposition and freckling. And then also, oddly enough, coincidentally, 5 to 10% of individuals with autism will have a large head. So they are shown to have mutations in this P10 gene, which is an overgrowth. And that could, I'm not sure why, honestly. I didn't have time to do all the reading and see if things are up to date. But it's probably due to overgrowth of the head and overgrowth of the brain is causing the autism phenotype. So what I want you to think about in these last few slides, and I would go over them before the test, is what testing you would do with what levels of autism and different things. Because we've gone over a lot of different types of tests, from the triplet prime repeat to targeted mutations, single gene, panels, exome, genome. You may want to know when you need to order what, okay?
So does the doctor order the exact test that you're going to do, or are they just like, here's autism and then you have some discretion?
We do not run a test without an order. The
So
doctor
like
has to tell us what to do.
Okay, so
So do you ever get a test order that you think is wrong, like they're looking for autism, but they order something like
Like a test for leukemia? Yes. But that was actually just a coding error. They checked the wrong box. Many labs have lab genetic counselors. We are trying to hire a lab genetic counselor. And what happens is the lab, if this is a career that some GCs want to go into, they vet all the tests that come into the lab, check all the orders, make sure the results correlate, if the physician has any questions, assist in reporting, write reports, sign reports. So it's a different, most people think of genetic counselors in the clinic seeing patients, but the lab genetic counselor is a really, really important aspect of it. We cannot cancel an order either.
but do you recommend ever, like if they ordered the wrong thing or if it, okay.
So especially at VCU, we don't have a very large test menu, so a lot of tests get sent out, and they all go through us for approval for various reasons. And we call the physicians and say, one time a physician ordered 2 panels for something, and 5 genes on one, 6 genes on the other, but 2 were the same. And one was a juvenile panel, and I'm like, is this an adult? You shouldn't order the juvenile panel. In the end, the doctor has the say. We're not mean about it, what we're thinking. But we do try and guide the decision based on the genetic knowledge. And the genetic counselors are better at that because that's what they do. But we do it.
If you find like a novel mutation or something like that, do you like that's a pathogenic mutation, do you send it to like a researcher to like be like you should look at this or something like that, or do you guys kind of publish it yourself and have people kind of
well,
figure it
that
out?
depends on the lab and who's running the lab and what their research interests in. Remember, I'm trained as a PhD research scientist, so I may have a small amount of protected research time, and if it's my interest, then yes, or if I'm collecting things. I may have a collaborative relationship with Rita, so I send her my patient, or we can't send them her patients, but we can tell the patient that there's a researcher who's interested if they're interested. It's the onus is on the patient to be interested to contact. We can give both people the information. And a lot of people do that. Some labs just don't have time, they're so busy. But in my mind, I want to do what's best for the patient. If someone's gung-ho interested in research, I'm going to find them a research project. And the genetic counselors do that too. A lot of what they do is trying to get people with resource groups or with
For the incidental findings, is it the clinical lab's responsibility or the physician's responsibility to tell the patient?
it is the physician's responsibility. My duty is to report or not. If the opt-out comes through on the consent, I mask and don't look. If it's there, I tell them. Remember, I just provide the report to the physician, and the physician has to give the information. Now, if they need help in interpreting the information, that's one of the other reasons why we're here.
Would your report say, like, say you were testing for muscular dystrophy or something, on the top, like, positive, like, and then incidental find, like, how would you, or it would be a separate report, like, how would you say, like, well, we also found the
I mean, if it were, let's say it were negative for anything, the result would probably be negative, comma, secondary finding positive. You know, it would be very clear. Anything that ends up, we assume that anything that ends up on page 2 will not be seen. I mean, it's just true. People don't have time. I'm sorry I took up some time in your review session.
I know it's all like sequence based. You guys don't do any like expression based tests or anything like that. Is that just because it's not as concise or the technology's not quite there?
The technology's there, but I don't think that the scientific knowledge clinically is there, because everybody's, you're looking at expression levels that are changing by some small fraction. We have no idea what they mean. One thing that the clinical labs are starting to do is if we find, not our lab, but I'd like to institute this as we go forward, is if we find a canonical splice variant, I would like to have clinically validated. Remember, all our tests are validated. A test that we could get an isolate RNA from the patient to show that that splice mutation in the canonical splice it results in an mRNA that is different, because that's another level of evidence that's truly pathogenic. And some of these genes that we don't know what they do, that's critically important. But we just don't have the time to do that. If you guys have any questions or anything, most of you know where to find me. My email is available. I will not be here next week.